Next on the program is a fascinating pharmaceutical research by the Orpheus group. Would you tell us the latest developments in pharmaceutical research relating to stuttering? And thank you for being here today.
Thank you, Ryan, for the introduction. Good afternoon, good morning, everyone. My name is Smiljana Ristic, and I work for a pharmaceutical company called, let me see whether I can share my screen. Can you see my screen? Can you see my screen?
Yes.
Good.
Yes. Yes.
So the name of my company is Noema. It comes from Old Greek language. It's a thought, a reflection, the original meaning. And the company is focusing on CNS, central nervous system disorders. Some of them are rare diseases, but some of them are diseases where we understand the mechanism of the disease, but there are no drugs available. And today I will talk about pharmacological therapy in stuttering, a little bit about history, what has been done so far. Then I will present our investigational drug, Gemlapodect. The main body of the presentation will be about the study that is going on now in USA and in Australia. And then I will conclude with prospects of the drug and the challenges and opportunities of drug development in stuttering. So the first slide summarizes a little bit what has been done so far. As you know, about 1% of adult people are people with stuttering, and it's acknowledged that it can have negative impact on social and occupational functioning. So stuttering is not what is called in drug development rare disease. It's fairly common. 1% is fairly high prevalence, what we call. Even though it's acknowledged that the prevalence is high and it has an impact on people's life and functioning, there are not yet drugs approved for stuttering. So you cannot go get prescription and go to pharmacy and buy a drug and read in the label of that drug that it's been approved for stuttering. There's been quite a lot of small clinical trials looking into different neurotransmitters and mechanisms, but it didn't work. For instance, the drugs that can control depression, like serotonin reuptake inhibitors, and also the drugs that work in anxiety, like benzodiazepines, they don't have effect on stuttering. The most promising area of research in stuttering, in pharmacological therapy of stuttering, is some kind of modification of neurotransmitter dopamine. Dopamine is well known to have central role in motor and cognitive function. And for speech, it's important this integration of cognitive and motor function is important for speech. It's been documented that the medications that increase dopamine levels, drugs like L-Dopa that is often used by people who have Parkinson's disease, those drugs that increase dopamine level are associated with worsening of stuttering. Consistently with that, there are several small studies that show that the drugs that lower dopamine 2 levels, what we call dopamine antagonists, they can have a good effect on stuttering. And the oldest drug tested in people who stutter is Haldol, very old drug, but also the new drugs, what we call atypical antipsychotic drugs like risperidone and olanzapine, they do have positive effect in stuttering. These drugs are often called antipsychotic drugs because the main indication are psychotic disorders. But those drugs have been tested in non-psychotic disorders like post-traumatic stress disorder, obsessive-compulsive disorder, and there were evidences that they can work in those disorders. Unfortunately, dopamine 2 antagonists are associated with significant adverse events, the most important being movement disorders, and these are involuntary movements, or metabolic abnormalities, obesity, impairment in glucose levels, and metabolic syndrome. And because of the side effects, they have never been developed for indication like stuttering. Stuttering is not considered to be as serious disease as psychotic disease, and you need to have a drug that has a better benefit-risk profile. There's been also a program with the drug called ecopipam. You have maybe haven't had a chance to read about that. It's an antagonist of dopamine 1 receptors. The drug was tested in a small open-label pilot study in patients with stuttering, and the people had improvement in stuttering. Medication was well tolerated, which is different from dopamine 2 antagonists. Then another study was done a few years to 3 years ago, a proper drug development study that treated ecopipam as investigational drug. It was a double-blind randomized control study, and we expect results to be published soon. So in the last 10 years, most research in a quest for finding a drug pharmacological therapy for stuttering is around dopaminergic transmission. The drug that Noema has is called Gemlapodect. It's an investigational drug, and it's currently being developed in 2 indications. The first one, as you know by now, is stuttering. The second indication is so-called Tourette's syndrome, a syndrome that has some similarities with stuttering, that it has speech, but also movement disorder symptoms. So Gemlapodect is an investigational drug that modulates dopaminergic transmission through the inhibition of a very specific enzyme that's called phosphodiesterase 10A. And this enzyme is expressed almost exclusively in striatum. Striatum is part of the brain that is important for this cognitive and motor interaction in an area of the brain that is called basal ganglia. The mechanism of this drug is such that it's the likelihood of metabolic and movement disorders that are a well-known problem with the dopamine 2 antagonist is less likely. Gemlapodect is an oral drug, so it's a pill, capsule, and it's suitable for one's daily administration, which is good. So basically it works, you have a scheme of dopaminergic synapses, and this drug works indirectly by reducing dopaminergic signaling at postsynaptic neuron level. This slide is just to give you a broader context of what drug development means and where we stand with Gemlapodect now. This is an example of typical drug in development. First, you have to do a lot of preclinical research in vivo and then in vitro in animals and show that your drug has a prospect of being effective and safe in humans. And then you start your clinical development program, which usually consists of phase 1, 2, and 3. Phase 1, the first phase of clinical development, is usually done in volunteers. Only in cancer it's done in patients because cancer drugs are quite toxic. So after finishing your phase 1, you know that the drug can be safely given to patients, and you have some idea about the dose of the drug. So then you go into your phase 2. Phase 2 is a small study, fairly small study, 100 to 500 patients. This study will have 60 patients only, where you are trying to test your hypothesis that the drug works in your selected disease area and that it is fairly safe. Once your phase 2 is finished, then the company makes decision. If phase 2 is positive, then the company decides to make the major investment, because phase 3 is very, very expensive. In phase 3 studies, usually more than 500 volunteers or patients are tested, sometimes up to 5,000. Some drugs even tested 10,000 patients, depending on the disease and the primary endpoints. And after all those years of preclinical research, which takes 3 to 6 years, and then 6 to 7 years of clinical research, if all fine, you can get marketing authorization to use your drug in a prescription, to buy it in a pharmacy. Even after marketing authorization, some additional work may be done, and that's what we call phase 4. So where we stand now with Gemlapodect is phase 2, phase 2B. So we are doing a phase 2 study. If that is positive, we will move to phase 3. Now back to our phase 2 study. This is the title of the study. It's a typical investigational study for investigational drug, which means this is a rigorous study. It's a double blind, which means that neither investigators nor patients know who is taking active treatment and who is taking placebo. It's placebo controlled, meaning that half of the subjects will be on placebo, not on the active drug. It's what we call phase 2B, so phase 2. It's multi-center study, international study done in Australia and in USA. For the time being, this is the first study in stuttering in human, and we decided to limit it to English, to one language only, to reduce the variability. It's a prospective study, and the goal of the study is to evaluate efficacy and safety of the drug, gemcitabine, in adult male patients with what is called childhood onset fluency disorder. Childhood onset fluency disorder is basically a scientific term for stuttering. We are recruiting only adult people because, as you know, most young people somehow grew out of stuttering. Among young people, about 5% of them can stutter, and in adults, it's only 1%. And we believe that the problem and the treatment should start in adults because they are more motivated, more willing, and they are less likely to spontaneously eliminate the condition. Male patients only because the drug had some evidence of teratogenicity. Teratogenicity means that the drug could have effect on unborn babies if the mothers get in touch with the drug. So for the time being, we are recruiting only male patients. In stuttering, more patients are male than female, as you know. So now on the next slide, we have presented the design of the study. Everything in the study starts with consent. So the subjects who are interested to take part in the study have to come to investigational site, and they hear a little bit about the study. They can ask all the questions they need to ask, and after that, they sign the consent that they want to take part in the study. After signing the consent, the study procedures can start, and then the investigators will do the screening. So the investigator will check all inclusion-exclusion criteria. They will do some lab analysis, ECG, to make sure that the patient is suitable for this study. Once the screening is finished and the patient is what we call eligible for the study, the patient will be randomized by computer. So the computer decides by chance who will get active treatment and who will get placebo. Equal number of people in this study will get active treatment, gemcitabine, and placebo. After the end of the treatment period, which is about 10 weeks, so 10 weeks is enough for this drug and for this mechanism of action to show efficacy, if there is, and we hope there will be. After the end of treatment, patients are asked to come back 1 month later, 4 weeks later, and then another safety assessment is done. They will be asked about adverse events, and also the lab analysis and ECG will be done. And that's basically the end of the study for the patient. Now, let's look a little bit into inclusion criteria. So we are trying to keep it as inclusive as possible so that we can find the patients and recruit them in a short time period. But also there are some limitations, and you don't want to recruit people who have other chronic diseases or therapies that may interfere with the study treatment. So we are recruiting male, as mentioned before, male adult people, age 18 to 55, body mass index between 19 to 35, which means that almost everyone can take part. Only people who are very obese, they cannot take part because of the distribution of the drug may be different and they may need different doses of the drug. People who can take part in this study should have a history of stuttering for 2 years at least before the study. And stuttering should be consistent with developmental in nature, meaning that the first symptoms appeared before the age of 8. The people who are interested in the study will be asked about their stuttering experience. And if they have moderate stuttering, they are eligible, entitled to go into the study. If they have very mild or extremely severe stuttering, they may not be the best candidates for this kind of treatment. So we are looking here for people who believe that their stuttering is moderate. Because the drug had teratogenic potential in rabbits, the patients who take part in the study, subjects must use condom during the treatment of the treatment period, but also for additional 90 days, which is one cycle of spermatogenesis. Subjects must discontinue all medications used to treat stuttering for at least 14 days prior to screening. You remember we said there are no drugs approved for stuttering, but some drugs are used for stuttering off-label, and these are the drugs that we mentioned before, D2 dopamine antagonists to dopamine 2 receptors. Other chronic therapies, for instance, therapies for anxiety, for depression, for allergy, can continue, but they should be stable, not change just before the study and during the study. We would not like to include patients who will be using cannabis or cannabis products during the study because it can interfere with the drug at the level of D2 receptors and also with the safety of the drug. So these are the key inclusion criteria. Then what is very important in every clinical trial where you're examining the investigational drug is what we call endpoints. How are you going to decide that your drug has effect? In this study, we have defined primary endpoint, secondary endpoints, and safety endpoints. The primary endpoint is change from baseline to endpoint in the scale that is called MLG-SSS. This is a new scale. It's been prepared for this just before this study, validated in a small non-interventional study. And I will show you a scale on the next 2 slides and explain why we believe this is the best scale in a clinical trial that tests pharmacological therapy for stuttering. The secondary endpoints are the endpoints that are commonly used in other studies in psychiatry, but also in allergy, in low back pain. So Sheehan scale is talking about functionality. How do you function in your daily life, in your work, in your family, in your social interaction? Then we have 2 scales where the participants will assess themselves in terms of change in the condition and severity of the condition. Patients will also be asked about satisfaction with the medication that they use during the study. So all the scales that are mentioned here that are printed in orange are the ones that are done by patients. So the data go directly from the patient into iPro. It could be iPod or it could be a mobile phone, and they go directly into clinical database. So the investigator cannot interfere with the patient's opinion. It's directly a patient's opinion. The scales that are presented in gray, so what we have here, clinician global impression change and clinician rated stuttering severity, they are done by the investigators. Global impression change is asking investigator whether he or she thinks that the patient is doing better or worse in comparison to the beginning of the study. SSI-4 is very typical for stuttering. It's what people call objective scale. So this is the scale that measures the fluency of the speech, that measures duration of stuttering and percentage of syllables that been stuttered. And that's done by one rater, centralized to reduce the variability. In some other studies, SSI-4 as objective scale was used as primary endpoint. But in this study, we have decided that the primary endpoint should be subjective endpoint. Because
Smiljana. Hi. I just needed to give you like a 30-second warning because we have to start our research symposium and we have to kind of reconfigure our breakout rooms. So if you have any final words, I'm sure everyone would love to hear it.
Good. Thank you, Tricia. So we do also the safety assessments, which are common like in any other studies. And I will just move now to the concluding slide. Gemlepodic as investigational drug has mechanism of action in animal studies that support efficacy. Phase 1 human study confirmed the dose and safety. Recruitment in phase 2 study is ongoing, and depending on the results of phase 2, phase 3 will continue if the results are positive. Thank you, Tricia. This is the end of my presentation.